LPXTG is there, right? That is the 5 letter amino acid. That is the identification for that. If that LPXTG domain is there, we can say that protein is like present in the cell wall anchoring the region. We can directly say that that specialization we can say, right? For those proteins, we can identify the motifs. So that is why we are going for to search the motifs if our protein is having any special feature, because actually if they are finding a drug for this LPXTG domain instance, they are targeting this LPXTG part, like targeting the LPXTG part, and the same thing we are finding out in some other species. It is also may have a it may also have the same property, because that is also from the same family of protein, right? Let's say the motif it is sharing, so the same drug can be applicable to this bacteria also. We can use that one. That's why we are identifying similarity between the proteins. So as we are evolving, right? Like all the organisms are evolving only by a minute changes. Like it is evolving. So for one thing applicable in a sense, it will be applicable for the other things also. In that concept only, we are going for identifying proteins, like motifs and domains, because the whole thing we cannot study. Some proteins might be like having a very bigger sequence of 2000 amino acids if it is having, and the same property it is sharing with the protein which is having 200 amino acids. But the 200 amino acids, they can easily solve the structure and they can easily identify, because it's the weight of the protein is very less, and while like macromolecular thing is tough to handle, right? So the thing which is very small in a sense, they can easily like connect with that thing. That's why they are checking for that specialized motifs. Now we will see some tools like how to identify motifs for our protein. Okay, I will share you. We'll first see about meme. Have you heard of this word, like meme suite? Meme suite. Yeah, this tool has been widely used for identifying the motifs. Okay, can you see this thing? Like this is the whole tool we can see. Actually, there are various options are there. You can see like how it is working. This is a flowchart, like how it is working. Like you will input your DNA or RNA protein sequences, right? So it will do a motif discovery using these things, and the discovered motifs will be highlighted, and it can be scanned for motif scanning through this is like for finding motif scanning is there, right? The sequence databases and motif scanning is used for finding similar kind of motifs which is present in the other species. So, and we can annotate the sequence, we can, and several things are there. If we are going for motif database, motif enrichment we can do, and go databases. Like this is a flowchart. If you do like study about these things, then you will get to know what are the things are there, and specialized other things are also there. They're telling you to, like we can check for find individual motif occurrence and the simple enrichment analysis, multiple EM for multiple location. So for you people like for understanding for the practical sessions, we'll go for meme suite, multiple EM for motive education. So illustration, this tool we are gonna use. Okay, then we'll see like how the data submission is gonna happen. Okay, whether we can use any kind of data, either DNA or RNA or protein or custom of alphabet sets, data sets we can use. And mode also we can use the classify mode, distributive, differential, enrichment mode. Each mode has different unique characteristics. If you click here at the question box, it is giving you. See, if you provide one set of sequence of meme discovers enrichment set of these things you can read, because this is just about how it is working. At your free time, you can read. And we are going to use like DNA sequences or protein sequences. So we are going with the default thing. And while inputting the sequence, they are giving you two options. Either you can upload the sequence or type in the sequence. Type in the sequence, if you give, it will open a box. We can paste our sequence. So like we have already, I'm already in that SF page. So I'll take the same sequence. Last class I have given you, right? I will give you the same sequence and find out any kind of motif is there or not for this protein. I don't think, because this protein is very rare and I don't think it will be there. Let's check it. Yes.
Can you paste this sequence in a chat box so that we could practice
Yeah.
simultaneously?
Yeah. Okay. Okay. Okay, then zero or none. Actually, I am pasting one sequences, right? So I have to go for any number of repetitions. Then how many motifs should be found? Fine, like I'm giving the value as a 3, and these things are like optional. Whether you want to give job ID and all, that is optional. We'll just check because this protein is not having much information. We'll see whether we are getting for this protein ID or we'll go for another protein ID. I will give you. You can search like patterns for those motifs for those proteins. Okay, we got the results. Okay, finally we got some results for this one. Okay, we have restricted to 3 motifs, right? That's why it's stopped after finding 3 motifs. Q, they have given you the width of the motif sizes. But actually, this is 31. 31 in the sense it will go under the domain region. The third one it is there, right? But I don't know why it is coming under this motif region. More number of oh oh. Okay, 342, 406. We can to 83 and 272 to 277 is on 5 regions and 342 to 372 and 406 to 306. This is the location in the whole intestines because this is 635 residues are there in this protein. It's around 635. So here they are giving you the motifs. What are the motifs are available for this one? Primary sequences, sequence faster protein, and total size it is there, and the values for frequency, how it is like each amino acid, like the frequency of those amino acids in the thing. Okay, then other settings what we have given, like the input we have given, right? That is a thing. We will go with another sequence. I will try to give another sequence. This is another protein of 1000. Let's check for this one. Any number of repetitions should be okay. Okay, we have got the results for this one. Actually, this is a insulin precursor protein. You can see the difference, like what are the motifs are available in that one. It is showing you 3 discovered motifs. That is logo they are giving, right? No, like 3 motifs they are showing 1, 2, 3 because we have restricted up to 3 only. So that's why it is going to 3. If you want 5, also you can do. And width of those motifs, you can see 8, 9, and 8. And if you want to submit or down, actually, what we will do with this thing, that's what the thing, right? Mm, Find out motif occurrences in sequence data, mass rank sequences, affinity group motifs. The here we will go for like FEMO or TomTom or MAST because why we are going to do this thing? Like we want to find out similar kind of motif for like any other existing species which is having the similar kind of motifs. We can check through this one. So then it will lead into another page called MAST. This is motif alignment and search tool. Here it will use us okay, submitted motifs. We have submitted the motifs. Then we need to give enter the sequences. Okay. Then start searching. We have already given the input. We have chosen one motif from the thing we have got. Like from main results, we got one protein, right? A set of 8 amino acids motif we got. We are giving input that as a motif in mass to find out the similar. Let's show the result and tell you. Okay, now can you able to see the difference? Okay, like could you understand? This was a motif we are given in meme. Like this was a motif we are obtained from meme. The same kind of like for this one we are going for a search in throughout the database. Like the database where we have chosen that one. Okay, I forgot to mention the database there. Okay, I will show you the database again if you go into that page. So here the IDs are there. These are from the database, and whichever the sequence which is matching with this one, they are highlighted here. So these sequences are highlighting. Can you able to understand what I am telling? Is it a clear a bit or it is still confusing?
Clear, ma'am.
Ma'am, are you comparing this motif with the another motif, or
We'll go for two sequences, right? There or not. That's what I'm checking now, okay?
so ma'am, in the same sequence you are trying to find out the motif?
Not the same sequence. These things are other sequences. Like, let me show you again.
Is
See,
it kind of like blast?
yeah, it's a kind of blast.
Okay.
Here we got one motif region, right? Can you able to see the motif region?
Yes,
So
ma'am.
here I want to find out for this. I actually I'm choosing this motif as my priority. So I want to find out a similar kind of motif whether it is present or not. So here I'm choosing two three things are there. Find similar motifs published in libraries or library you supply, or find motif occurrences in sequence data. Rank sequences by affinity groups of motifs. Here I'm choosing this mass because mass is having the multiple like it is choosing a wide variety of things are there in mass. That's why I'm choosing for going for mass. So if I give mass there in data submission form, can you see I am submitted the motifs? Okay, I submitted one motif that is a
Yes,
protein, and the database I am choosing right here. I forgot to tell you the database what I am choosing. So if I want to choose GenBank bacteria genomes and proteins, wherever I want I can choose or other databases. The database which is like older than this thing. Okay, we'll go for GenBank bacteria and genome proteins, and version 210. Like these are all default. It will come and bacteria. If you want to choose any specific species, also it is available. I have given you bacteria, right? Okay. Bacteria. The genome database related to gene bank bacteria, genomes and proteins. So it will be related to the bacteria only. Will randomly
ma'am.
give Bacillus
How many data
available
sets
there.
are available
More
there?
than
Yeah.
10 database
Other
for
than
ensemble, ab initio predictions, and ensemble fungi genome and proteins. Like many are there. Some 10 to 15 are there. Can you able to see the thing? Like I am showing you. Can you able to see the thing?
No, ma'am.
no, ma'am.
No.
No, ma'am. It is not showing.
Oh, why it is not showing? Okay, a pull down menu is there, right? Can you able to see?
Ma'am,
Uh,
gene bank bacterial
yes.
genome and protein
If
is
I
not
will
available.
change now, you can now is it seeing you can change that ensemble maze. It has been changed because that folder menu is not showing in that thing. Mm, that full down menu it is not showing. So now again I will change it to gene bank and bacterial genomes. Is it like can you able to see because around 15 things are there.
Yes,
While
ma'am.
you
Got
will
it.
go to that thing, you can understand. And then another pull down menu at the drop down it is showing right like what is that name? Acariochlorosis marina, like that was like it is there next pull down menu, right? If you click that one, there it will show you the bacterial list what it is having. I will change it to Bacillus. Bacillus cereus I'm changing. Then default 210 will come. Now we have to start searching whether in that bacillus whatever I have given is there or not. Yeah, like I have chosen the database which is having genome of gene bank of genomes and proteins, right? And in that one I have chosen the family of bacillus series. So in that whole thing, this kind of motif is present in these regions. So from for this one, like GI accession ID they are giving, right? This GI ID we can see, get it in GenBank. So if you see, this is camelized. The first one is actually na. The problem is it is showing camelization. It won't show, I guess. If I can you able to see any pop-up menu there? No, na.
Okay.
Any pop-up menu is there? No,
No,
right?
ma'am.
Okay, because
Can you present in the entire view screen?
okay, let me show in entire view. Yes. Yes. Okay, now could you able to see the thing? Pop-up menu is there or not?
Yes, ma'am.
Yeah,
Camilla.
camelizing bacillus is there, right? And if I click the next one, that is from petropactin and biosynthesis of ABS, another protein. Like that is also from bacillus series. So the different proteins from the same bacteria is having a similar kind of this motif. Could you able to understand now? Like this motif region, it is present in the bacillus species. Okay, SLS species with the different like various proteins is having that kind of motif. This is small d protein converting factor subunit from Bacillus series. This is what we are predicting. Like these regions of those the regions which is having a similar motif region which is present in to our like query one. Is it clear how to perform like
So, ma'am,
PM and master?
in the
Tell me.
same bacteria, there are different proteins. So you are
Yes,
finding the same motif in a different kind of proteins.
yes, different
That
proteins I am finding out, but those different proteins are there, right? The because of the same motif, it might have a similar property.
okay.
If it is binding to some other host, it may bind in a similar pattern. Now you are clear?
So the property of the motif is also same in a different proteins.
Yeah. Like if I am having this motif all over this region, which means like here also it is having, here also it is having the same motif.
Mm-hmm.
Mm. Then the property will be the same for this region because the continuous pattern of that amino acid is there, right? Let's see, Q Q Q L R W W I Q. Maybe this continuous pattern might have a particular function. That function can be we can see in all the proteins. Either it may be like a pathogen. We can like all the functional things are not pathogenic, right? So we are only thinking in the way of pathogenic things. Like, for example, if it is secreting some insulin also, then this region is responsible medicine. So all the bacteria which is having this region might be responsible for like producing insulin at this region. It's clear. Like, can you able to visualize that
Yes,
thing?
yes, ma'am. Yes, ma'am. Got it. So it is depending on the region specific.
Yes, depending on the region, and you can see like it is present here in 150 to 159, but it is present here in 95 to 102. The position may vary. That position may vary according to the species and its sequence length. Okay.
Yeah, ma'am. Got it.
Yes. This is how we have to perform new and master. Okay. To identify your like motif region of your protein. Then now I will give okay. I will give you one sequence for mm, this one. Then you can perform that one and you can see. Okay. I will give you an insulin receptor protein. Try to create the mass alignment. Okay. Just check whether you're able to do or not. After finishing, you just tell me. Okay. I'll give you time. Just try to perform.
Ma'am, it is done. Can I present it?
Yes. Yeah. Wait for a minute. The job has not done yet. Okay. Job editing. Go to the
Yeah.
first one. No, no. You click in another format. Go
Yes,
for
ma'am.
HTML format.
Yes, ma'am.
First one. Okay. So
Yeah.
now you can able to see now the motives and
Yes, ma'am.
let's proceed and change the database. If you want to change the database, you can change it.
Ma'am, can you go with the ensemble? Genome
Yes,
and
you
protein?
can go with genome ensemble.
Okay. Yes, ma'am.
And if
Got
you
it.
there, okay. Now you are understanding the thing now, like how it is working.
Yes, ma'am. Actually, what I observe here, ma'am,
Hmm.
in the same sequence, we can find the same motif
Similar
on,
motifs.
yeah,
Yes,
on two
that is
different
also
positions.
possible.
Okay. So how many maximum I can find it and minimum
Hmm.
number of motifs?
Number of
How many number of motifs, minimum or maximum, I can find in the same protein?
in same protein. That depends on the protein. Like we cannot predict the same thing now because some specific motifs are present only in the one region. But for example, if you if you think about a human protein, you like if you think about a hemoglobin, like it
Hmm.
may the gene sequence pattern is there, right? How it is arranged, like so, how I can say, mm, like let me take an example. Wait for a minute. I'll think about the example. How to say that one. See, not for producing one enzyme, not only one gene is responsible. Multiple gene can, like code for a similar kind of protein that you know, right? Like multiple gene involved. So wherever the multiple genes are there in the sequence, then we can find out the similar motifs right over there. Are you clear now?
Yeah,
If
yes,
you are
ma'am. Got it.
having gene pattern of, if you are having a gene pattern, think about, you just imagine we are having five proteins. Sorry, five. What is that? 5 genes which is responsible for coding one protein. So
Yeah.
wherever that five different genes responsible for one protein. So wherever it is present, we can find out the similar motif in genes. Okay, that's how the repetition of the things are there, like seeing the sequence.
And ma'am, what this E value is it? Is my cursor is visible, ma'am?
Yes, but it's okay. You just tell. I will try to view it.
Yeah. Actually, if we see the E value is increasing, okay,
Yeah, yeah. Like if
from
the
top
e
to
value
down.
is
So what does it
zero,
mean?
that is having the exactly matching query sequence. It's exactly matching with the query sequence. E value that according to each tool that differs actually. So in mass, I will also see. Wait a minute. E value. So if you see the top scoring sequences are there, right? The above thing, the black color thing they are mentioning, right? Each of the following sequences of e value is less than 10. So whichever is having the e value less, actually, e value. If you see the blast results, the e value there it shows like if it is having e value is 0%, then identity of the sequence will be more, right?
Yeah.
Likewise, here also, the e value whichever is having less than 10, they are considering only that sequences because that might have a similar that might share a same thing. That's because it's all about algorithm, right? Everything is predicted based on numbers. Some
Ma'am,
I guess.
ma'am, but you said that the same motif, this this same motif is finding in the all these regions, right, ma'am?
Yes.
Same sequence. So, so how this is very no
How this will vary?
no, ma'am. If we see in this sequence, then
Yes.
here it start from the E R C W T, but in the
Hmm.
downside sequence it is starting from the high R C W T T S.
Is it
So
so? Let me check it.
yeah, that's it.
Yeah,
Ma'am.
yeah. Now, okay, okay. I can understand your question, I guess.
Yeah.
Okay,
So,
that highlighted region. Tell. First tell.
ma'am, there are two motifs in the same
Okay.
sequence, okay,
Hmm.
and each time the sequence is varying.
Hmm.
Can you see
It's
it here?
yeah, yeah. I can see that the changes there. Like in one thing, it starts from for my protein. It starts from can you click the sequence at the down? Like there is an option there, right? Like
This
down arrow at the same line.
this one.
Move your cursor. Yeah, I click that one. Now we can
Mhm.
easily see the things changes, right?
Yes, ma'am.
Yeah, it starts from one pattern, and for other thing, it is following a different pattern, right?
Yes,
And you
ma'am.
can see the highlighted things like Q, which like for something the E value is 1.3 E7. The propensity, I guess, like it's because yeah, actually, wait, I will also think about this one. Whether I don't know whatever I'm saying is a proper answer or not. I will look into this one. But according to my like understanding, this is a thing. Like the amino acids are there, right? Which might share similar properties, right?
Ma'am, is it
Both will
some
settle. Okay.
ma'am, is it something like blast that if our E value is less, then it is more similar to our first motive region.
Yes, that's
Whatever
what.
query we have given here,
Yes,
right? This is
yes,
our query sequence, and this is our output sequence. So this query will be same as will be same with our outputs, right, ma'am?
yes, yes. The same thing we'll do for blast, right? That's what the
Yeah.
e-value mentioned. Where the value more, the value lesser. Like if the value is at the zero, like value is very less, the identity will be more.
More.
So
Yeah.
here it has
So,
choosing only the e-values which is less than 10, not more than 10. If it is crossing more than 10, then that will not be a proper similar matching pattern.
hmm.
Mm. That's why the value is less than 10. And I will look into this one, like why the e-value is decreasing or not, and then I will let you know in next class. Okay?
Ma'am, what I'm thinking that this is our query sequence, right?
Yes.
This one. So it is more identity to sequence, the output motif sequence, right?
Yes.
Something. And each and every time the sequence identity is decreasing from
Then that's
because
why
of here,
the pattern is also changing.
that is why pattern is also changing. Yeah.
Yes.
Okay.
If it still didn't clear also, then I will try to explain more in the next class. Okay? Like why the algorithm of this one.
Okay, ma'am.
Okay.
So let's stop the sharing.
Yes.
Thank
Yes.
you.
This is clear, right, for other people?
Yes, ma'am. Yes, ma'am.
Okay. Okay, you save this result like the question, the query I have given you, right, for your record thing, and like incorporate meme and mask and give you the interpretation. Okay.
Okay, ma'am.
Then the same thing you can do for using five blast also. Like five blast, you know, right? Okay, five blast. Okay, I will go for five blast. That is also a pattern finding thing. There we have directly did the blast with mask tool, right? Here if you want to find out in any other different database, like here you are having a different set of database, there we are having ensemble database, those kind of database we are having there, right? So if you want to do again like when you plot or something, then we can try it here. So what the thing is, you have to click five blast at the algorithm, at the end there is an algorithm, right? Oh no, here you have to give pattern, not like okay, this is another thing I will, this is pattern we have to give. We should not give here it as a motif region. Motif it won't take. Okay, first we will find out a pattern. I will show you how to find out a pattern for a protein using. Okay, you guys know about patterns, right? Um, Already we have discussed, I guess. This is how. Okay, let me show whether it is there or not. So, can you able to see this thing?
Yes, ma'am.
This is called what we will call as a pattern. So this DNG means like aspartic acid and glycine will come, and any amino acid can come, and M or V, methionine or valine will be there. In the next position, alanine or leucine will be there. Like this is how we will create a pattern with the help of many sequences. We'll try to create one pattern, right? This you have studied in your class itself, right? So first we will try to find out a pattern for our interested protein sequence. Then we'll try to find out with the help of patterns. We'll try to find out similar kind of proteins whether it is there or not. Okay. So for doing that, we'll use a tool called ScanProsite. ScanProsite will not give you pattern. Prac will give you pattern. Okay, we'll perform both like what is that? Mm, Prac and as well as Prosite scan. We'll check ScanProsite. This scan process tool is I think handled by XPC tool, like XPC. Here we can find the motifs for our sequence, and here also the same thing we have to do. What we have done in that other tools also the same thing. We have to paste our sequence and identify the motifs, like whether it is there or not. Okay, I will use the thing insulin precursor. Uh, I have pasted the sequence of
Ma'am, can you paste the sequence in your chat box, or are you using the same sequence?
yes, one second. I will give. Is anybody is performing, or should I? Okay, I will tell the thing. What are the things in results? So we have given input sequence as IGF-1 or human. This is the insulin receptor protein. One second, I don't forgot the name. Insulin like growth factor receptor 1. That was the protein we have given here. Now we can see it's a sequence of around 1 to 367. Okay, now we can see what are the things it is having. It is having an FN3 region. If you click that region, like that will show that is a fibronectin type 3 domain profile. So like profile, like it will, if I click that region, it will move on to another page. I will share that page. Okay. If you see here, fibronectin type 3 domain profile. This is what that description about. They are giving the description about those things, and at the end you can see, like while moving down, you can see the things, like what are the things are there and who are all like published those things. Those details are there. The profiles I couldn't see. One second. See, from 491 to 609, they have predicted this as a like a domain region of fibronectin type 3. And for other things, like continuously they are having FN3, right? FN3, FN3, FN3 as a fibronectin domain. And this is the last one they are mentioning this as a protein kinase domain profile from 1999 to 1274 of this amino acids. On binding side, the details they are given, and you can see which were patterns in the sequence. These things are like by this way also we can predict the domains for our protein sequence. Can proceed. I have given another sequence, right? In that one, this is just an insulin protein like this. Oh, okay, okay. This is human insulin, like 3. That was the protein I have given the next time, and I am predicting one like a pattern, like 131 amino acid at the end region. It is having like 115 to 129. So if I click that region, it will go into another page that is called as insulin family signature, and details they're having where the insulin family proteins of groups 1 are related to what those descriptions are there. And technical section, if you see, they are giving you process method with tools information. The insulin family signature pattern also they're giving. So CCTP, this is the signature for this protein. I can do blast for this pattern, copy, and blast.
Ma'am,
Yes,
ma'am, from here the pattern expression only we need, or some
only
other
expression
output
is
we
there.
need.
Nothing is needed. Only that profile of that
Okay,
the
this
expression is
concession pattern expression that only
yes.
we need, ma'am.
That only will need, not more than this thing. And one second, wait a minute. I'm sorry. Uh, okay, this thing we need, like this pattern. That is the only thing we need in this one. Nothing else.
Which ID we have to use, ma'am?
ID sequence. Okay, I will give you the ID. Okay, 1 second. For scan proceed, you can use this one.
Okay, ma'am.
And if you want to do any other 14 also, that's fine. That's up to you. Or else you can use the same thing. And another tool is there, right? Prat, I have told you, right? That is also like used for discovering patterns of conserved in a set of 14 sequences. That is also we can check it. Prat tool. And here we can share tab. This is an XPC tool actually. We here we can find out a pattern. Wait, let me. You can create your own pattern also here. Give some multiple set of sequences and try to create a pattern for that one. If you have an alignment file also, you can give and get the sequence. I will paste the same sequence what we are using for the last thing and I will check it. Starting, we are generating a pattern, okay? In that process, we are just getting a pattern of the general ones. But in this track tool, we are generating our own pattern of pattern for our required sequences. So I need to paste two sequences. I'm pasting two sequences of insulin from different organisms. And parameters, if you want, you can change it. Actually, the pattern parameters I'm not changing. I'm going with the default thing. I want to be at least 100% to sequence and maximum length should be 50. This is you can check. Then run plat. To be able to see the things like the pattern it has been created for the input sequence we have given, right? This is what the pattern we are checking. And the thing we can do is start the scan. If you see, it like we were already in the prat tool. Now it has been moved to after generating the pattern. It has been moved to process scan tool. Now at the end you can see start the scan. Now if you apply this one, it will help us to find. It will help you to find out the similar things, similar patterns. Okay, I got two similar set of patterns. One is from probable insulin like one is from Gospela Millennium Booster, another is from Homo sapiens. Actually, this is the same thing I have given exactly. But this is how you have to perform your things. All the things like just identifying motifs and patterns. It's the same thing, repeating, we are repeating again and again.
Ma'am, can you give IDs for this?
Yes, okay, I will give you. Mm. Two, two things. Okay. I will type it. This is can fifth ID, I guess. One second. We are performing name, mask, scan, prosite, and prep. Okay. Put everything in one under one experiment. I don't know how many experiments we finish. We just put one like experiment number as the pattern and motif prediction, sequence pattern and profiles. You put it as sequence pattern and profile, and inside that you just give A as mem or mask, and B as camposite, and C as prat.
Ma'am, could you please repeat the title?
Yeah. Okay. Okay. Put the title as sequence patterns and profiles, and inside that A, B, C you just mentioned, and mem and mask just camposite and prat.
Yes, ma'am.
Yes.
So
And
three sub topics, right, ma'am?
yes, yes, three
Okay.
subtopics. Then protein motifs domain analysis is there. Okay. Okay. Only less content are there. Like almost we have covered most of the things. The rest of the things we will see in the next class. Okay. Okay. Also didn't show their records. What about Satish? Did you finish or not?
I have finished.
Okay. Chandani, you?
Yes, ma'am. 8 experiment I have finished.
Yes. Okay. 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 I finished. Yes. I asked Chandani, but Chandan answered it's fine. But again, I will ask you, Chandani, you did you finish? Okay, try to finish as soon as possible. Okay, and finish this movement match also. This experiment also will finish. In the next class, we will see domain analysis. Then we'll finish like one, two, three things are there only. Then we will see all those things. Okay. Shall I wind up?
Okay,
Okay.
ma'am. Thank
Yes,
you.
Yes.
ma'am.
Thank you.
Thank you, ma'am.